Welcome to another lecture by MedicoMedics. Chapter 6, the complement system. In this lecture, we will talk about the complement system, discuss its pathways, some key steps involved, its functions, regulation, clinical relevance of the complement system, and end with a summary. Now then, let's talk about the complement system, which is an essential part of our innate immunity. Now, it is a collection of over 30 proteins found in the blood plasma and on cell surfaces. These proteins work together to complement or enhance the actions of antibodies and immune cells in fighting off infections. The key functions of the complement system include, number 1, opsonization, so marking pathogens for destruction. This means tagging or coding pathogens with complement proteins. So we have a pathogen, and we will coat this pathogen with complement proteins, especially one called C3b. What this does is it marks it now. Essentially, there is a flag planted here on this pathogen. So our immune cells, or phagocytes like macrophages and our neutrophils, essentially our immune cells, they will then easily recognize this marking and come to destroy it. So that's opsonization. Second, we have pathogen lysis. So complement proteins can form what's called the membrane attack complex, or MAC. And this should not be confused with the computer Mac. Now, this complex punches holes in the membrane of the bacteria, especially in gram-negative bacteria, leading to cell lysis and cell death. And we're going to deep dive into the membrane attack complex in coming slides. Then we have number 3, recruitment of immune cells. So small fragments like the C3a, for example, or C5a complement proteins, they act as chemoattractants. So they call in neutrophils and other immune cells to the site of infection, helping mount a rapid inflammatory response. Now, clinically, deficiencies in complement components can lead to serious infections or autoimmune diseases. We're going to discuss this as we move along in the lecture. So overall, the complement system isn't just a backup. It's actually a frontline defender that tags, attacks, and recruits to keep infections under control. Now then, let's break down the complement cascade pathways. So you can follow the illustration here. We have our classical pathway, we have our lectin, and alternative. One thing to keep in mind is that the end result of these different pathways is to arrive at lysis, more specifically microbial lysis. So we begin with the 3 distinct pathways that initiate this cascade. First, we have the classical pathway. And the classical pathway is triggered by antigen-antibody complexes. So this activates a complex of proteins known as C1q, C1r, and S. And for your information, antigen-antibody complexes refer to clusters formed when antibodies, usually immunoglobulin G or M, bind to antigens, marking them for immune recognition. And these complexes trigger the classical complement pathway by activating, for example, C1q. Next, we have the lectin pathway. It is activated by microbial carbohydrates, specifically when mannose-binding lectin, or MBL, binds to pathogen surfaces. This activates MASP1 and MASP2. Finally, we have the alternative pathway, or the third route is the alternative pathway. And it is initiated directly by endotoxins or bacterial cell walls. And this leads to the spontaneous activation of C3. Further followed by the involvement of factors D and B, forming the enzyme C3b BB. Now let's pay a bit closer attention to the illustration right here. So despite their different starting points, right, you can see the difference between the classical, lectin, and alternative pathways. Despite their different starting points, all three of them converge at a specific point. It is the activation of C3. See, in the classical and lectin pathways, activation proceeds through C4, C2, which together form C4b2A, which is a C3 convertase that cleaves C3. In the alternative pathway, C3 is directly activated by C3b, which then participates in forming the C5 convertase. So just to clarify it a bit, we said, despite their different pathways, right, they all converge at C3. Once C3 is activated, it leads to the formation of C3b, which then participates in forming the C5 convertase. Now, in the classical and lectin pathways, this is C4b2A3b. I wouldn't say that it's necessary to know it in this great detail, just understanding that there are three different pathways. They all converge at C3. That's very high yield. They all continue to C5. And these C5 convertases then cleave C5, initiating the terminal pathway. So C5 activation ultimately leads to the formation of C9, which forms the membrane attack complex and causes cell lysis. So whether through the classical, lectin, or alternative entry points, all roads lead to C3 and converge to destroy invading pathogens. So just for clarification, let's review the key steps in the complement cascade. Like we said, initiation is what starts it. So one of the pathways is activated. Then we have C3 activation. You remember all these three pathways converged at C3. So a central step where C3 protein is cleaved into C3a and C3b by the enzyme C3 convertase. Now, C3b binds to pathogen surfaces, marking them for opsonization. If you remember from the beginning of the lecture, we talked about opsonization, so marking pathogens for destruction. So this is the key role of C3b. You can also follow here in the illustration. We have C3b here with a pathway to opsonization for the aim or purpose of phagocytosis. What about C3a? It functions as an anaphylatoxin or anaphyltoxin, so promoting inflammation, right? It attracts immune cells, promoting inflammation by recruiting immune cells like mast cells and neutrophils, and increasing vascular permeability. And you could probably already assume some pathologies where these different complement proteins are more present or relevant. For example, in pathologies related to autoimmune diseases, for example, where we have an ongoing inflammation happening. But more on that in coming slides. Then we have another key point, which was C5 activation. So we said the Three different pathways converged at C3. So this is super high yield. Then we had the continuation to C5. Now C5 is cleaved into C5a and C5b. So whatever is happening to, or whatever is high yield and relevant to C3 activation is similar to C5. And as number 5 is preceded by number 3, you can kind of assume the order just as a way to remember it. Now, C5b initiates the formation of the membrane attack complex, the MAC. You can see that illustrated here. Now, MAC creates pores in pathogen membranes, leading to their destruction. Beautiful. And here we have an illustration of the formation of the membrane attack complex. And the membrane attack complex, or the MAC, it's formed by complement components like C5b, C6, C7, C8. You can see multiple C9s. And its role, what is the role of this MAC? It's to punch holes in the membranes of pathogens, leading to cell lysis, so cell death. And why are we talking about it in relationship to C5? Because C5b is the initiator of this MAC formation. It binds C6 to C9 and anchors the complex to the target cell membrane. Excellent. Now let's just revise one last time. So the complement system plays a vital role in defending the body against infections, and it does so through 4 key mechanisms. We mentioned opsonization. So C3b coats the pathogens, marking them easier for phagocytes to engulf. Essentially, this makes the pathogens easier to find and destroy. Then we have chemotaxis. So complement fragments like C3a or C5a act as powerful chemical signals that attract immune cells to the site of infection. Then we had pathogen lysis. So the terminal components of the cascade, especially C5b, assemble to form the membrane attack complex. And this structure inserts into the membranes of bacteria, especially gram-negative ones, creating those pores that then lead to cell lysis and death. And finally, immune complex clearance. So complement help remove circulating antigen-antibody complexes that could otherwise deposit in tissues and trigger inflammation. So by binding to these complexes, complement allows them to be safely cleared by the spleen and liver, helping prevent immune-mediated damage, like, for example, that seen in diseases such as lupus. Now that we've seen how powerful the complement system is, it's important to ask now, how does the body keep this system under control? Because while complement is essential for killing pathogens, if it's left unchecked or not regulated, it can actually damage our own healthy cells. This is where regulation comes in. So why is it important? Simply put, to prevent host tissue damage. So we have to keep in mind that an uncontrolled complement activation can indeed lead to inflammation, cell injury, or even autoimmune diseases. Now, to avoid this, the body uses these regulatory proteins that act like brakes on the system. So let's go through the key ones here. We have CD55, also known as decay accelerating factor. Now, it blocks the formation of C3 convertase. And this, if you remember the cascade pathway, this will stop the cascade early and prevents excessive opsonization or downstream effects. Because if you remember what was used for opsonization, we had C3A and C3B, right? If we can somehow stop C3 convertase forming, we will then decrease the complement proteins here. And if we don't have these, we can't then have the effects of opsonization. Essentially, we can't cleave the C3, which would then cleave into C3A and B. Then we have CD59. Now, it plays a different role. It prevents the assembly of the membrane attack complex, or MAC. And this protects our own membranes from being punched full of holes. So we want the buildup of MAC, right? Or the formation of MAC, which came from C5. However, we want it on bacterial cells or pathogens that we want to essentially kill. We don't want them on the host cells, so we don't want them on our own cells. And this is the role CD59 here plays to prevent MAC forming on our cells. Great. Now after that, we have factor H and factor I. These work together to regulate the alternative pathway. So they inactivate C3b on self-tissues, preventing accidental complement activation against our own cells. Now, clinically, deficiencies in these regulatory proteins or these regulators can have serious consequences. For example, CD55 and CD59 deficiency leads to paroxysmal nocturnal hemoglobinuria, which is a condition where red blood cells are destroyed by unregulated complement at night, so nocturnal during the night. So while the complement system is a powerful weapon, right, these regulatory proteins are just as critical. They act as protectors or safeguards to ensure that the immune system fights pathogens, not the body itself. Now then, let's take a look at the clinical relevance of the complement system. So let's begin by taking a closer look at C3 deficiency and why it could lead to serious bacterial infections. Well, we remember C3 sits at the center of all three complement pathways: classical pathway, lectin pathway, and alternative pathway. They all converged at exactly C3. Now, without C3, the entire cascade is disrupted. And most importantly, in this case here, you will lose C3b. So you remember C3a and C3b. Now C3b was critical for what? For opsonization, so marking the pathogen for destruction. And without C3b, well, the process is going to break down, meaning that the immune system will struggle to clear bacteria, especially bacteria that are encapsulated. Now, if you lose C3, you also lose C3a, which was a chemoattractant that helps recruit immune cells to the infection sites. So now we are left with an issue here. Not only are the bacteria more difficult to find or spot because we don't have opsonization anymore, but we're going to have fewer immune cells being called for help because we've lost C3A. And if that wasn't enough, here's another thing. Without C3, which was a precursor to C5, right? So on top of all of this, C5 convertase can't form without C3b. So the terminal pathway is now blocked, which means what? Well, C5 played a crucial role in the formation of MAC. So it means we won't have any membrane attack complexes to lyse the gram-negative bacteria. So in short, we have no tagging, we have no recruitment, we have no lysis. And that's why patients with C3 deficiency face current severe infections, often with encapsulated or pyogenic bacteria. All right, so that's C3 deficiency. What about MAC deficiency? So in MAC deficiency, or C5 to C9, the complement system can't form these membrane attack complexes, right? And what's its relevance to bacterial infections like the Neisseria species? Well, the MAC complex is crucial for punching holes in bacterial membranes. We know that now. But due to the deficiency, we don't have that. And gram-negative bacteria, they rely very heavily on MAC-mediated lysis for its clearance. Good. Now, what about overactivation? So uncontrolled complement activation can lead to what? Well, it can lead to host tissue damage. And where do we see this? Well, in autoimmune diseases, like, for example, lupus, where excessive complement contributes to inflammation and organ injury through immune complex depositions. So as you can tell, it's a very interesting but complicated system to keep in mind when we're studying immunology, because we're talking about a system where if it's deficient, we're going to have potentially very severe consequences. But on the other hand, if it's overactive, we could have the same potential outcome. It's going to look different, but they both fall under pathologies. So let's say we're dealing with an overactivation of the membrane attack complex system, where it's attacking our own cells. Essentially, what is the potential clinical relevance here? Well, we have, for example, a drug called Eculizumab. I don't know if that's the correct way to pronounce it, but let's go with it. Now, it's a monoclonal antibody that binds to complement protein C5. And you already know now the role of C5 for the formation of MAC. Now, what happens is the drug will block or prevent C5 from being cleaved into C5a and C5b, of course. So the end result would be that this would block the formation of MAC. So this is going to be a useful drug because it stops downstream complement activation, which is critical, right, in diseases where the MAC damages cells, like the one we mentioned before, paroxysmal nocturnal hemoglobinuria, or PNH. Here, in this specific pathology, red blood cells lack protective proteins. They lack CD55 and CD59. And if you remember from the previous slide, these were our regulatory proteins. So when we don't have these, when the red blood cells don't have these proteins, it's going to be vulnerable to MAC-mediated lysis. And we don't want the MAC system to destroy our red blood cells. So we give this drug to stop that from happening. So in summary then, the complement system is a crucial part of our innate immunity, enhancing immune responses through opsonization, chemotaxis, and pathogen lysis. It works through three pathways. We have the classical, alternative, and lectin pathways, and they all converge at C3 activation. Proper regulation is essential to prevent the damage to the body's own cells. And that's the end of this lecture. Continue now to Chapter 7.